Hello, everyone. I'm Nadia Selami, PacBio segment lead for cell and gene therapy, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the use of HiFi sequencing to design and discover AAV capsids. I'm joined today by our wonderful speaker, Irvin Garza, who's a graduate student at UT Southwestern in Steve Gray's lab, who'll present after this introduction. We have a lot of great material to cover today. Both my introduction to HiFi sequencing for gene and cell therapy and Irvin's presentation are pre-recorded, but will be followed by a live questions and answers session. Liz Tseng, the Associate Director of Product Marketing for RNA and Gene Therapy Applications, will also be joining us for the Q&A. You are welcome to submit your questions at any point during the webinar by typing them in the area provided on your attendee control panel, and we'll get to them either in the chat or at the end. We've also uploaded complementary literature to your control panel, so please feel free to download this and take it with you. We will be recording this webinar and making it available for download in the next few days. Please keep an eye out for a follow-up email with the link to the recording. Also, immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. You can stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. So with that, let's get started with a brief introduction. I will start with a general introduction on AAV vectors and the tools that PacBio offers to support your gene and cell therapy research. Adeno-associated virus, AAV, is a common vector for gene therapies. The wild-type AAV vector genome is about 4.7 kb long and is packaged into a capsid as shown on the left. The AAV genome contains two inverted terminal repeats that flank the rep and cap genes. These two genes are essential for replication of the genome and encapsidation, with the cap gene encoding the proteins that make up the capsid. To use AAV as a delivery vehicle in gene therapy, you can construct a genome in which you replace the rep and cap genes with your construct of choice. These constructs can be packaged into capsids and then delivered to subjects. You can use PacBio HiFi sequencing along the development process of your AAV vector from capsid discovery through vector design to evaluation of your AAV vector preparation. The principle of HiFi sequencing is the ligation of SMART Bell adapters to double-stranded DNA, followed by the sequence of the circularized DNA template. The polymerase will pass the sequence multiple times, and the consensus sequence from these subreads is called a HiFi read. HiFi reads have 99.9% accuracy, which is Q30, and can be up to 25 kb long. For customers who want to take advantage of the long read length and the high accuracy of HiFi sequencing to analyze full-length AAV genomes from ITR to ITR, we offer an end-to-end workflow that is enabled on the Sequel IIe instrument and now also the higher throughput Revio. This workflow allows you to identify and characterize impurities such as truncated genomes or host cell DNA in your AAV vector preparation. The workflow is rounded out by data analysis tools that are available on GitHub or through the FormBio platform. Today's webinar is focused on using HiFi sequencing to discover and design novel capsids. Discovery and engineering of the AAV capsid allows for better targeting to specific tissues, lessening the effect of AAV on the immune system, and creation of a more stable AAV vector to help you optimize and scale manufacturing. Here's an example from the literature where PacBio HiFi sequencing has been used to find novel AAV capsid variants in human tissues through targeted sequencing of long amplicons. This allows the profiling of variants in high resolution without having to reconstruct the genome or cap gene from short reads, which enables researchers to accelerate the engineering and discovery of novel capsids for tissue specificity. The authors of this study noted that other long-read sequencing technology doesn't have the accuracy to be able to identify new strains or variants. Similarly, short-read sequencing doesn't have the read length to allow you to do this. PacBio's targeted sequencing portfolio includes amplicon sequencing as used in the study I just mentioned, and target enrichment. This is a cost-effective way to utilize the benefits of HiFi with the ability to customize for any scale project. For amplicons, you can sequence full-length PCR amplicons, even long-range amplicons that are 10 kb or longer. If you can amplify it, we can sequence it in a single read, giving you really clean data that is phased across the entire amplified region. This approach is really cost-effective for single genes or small panels, and PacBio has supported barcoding products and protocols for you. We also offer HiFi target enrichment with our partnership with Twist. Twist probes give very high capture efficiency and exceptional uniformity, while PacBio HiFi reads and their superior mapping give high accuracy variant calling, including structural variant calls and haplotype phasing. A great use case for target enrichment sequencing is to assess unintended AAV integrations as shown here. The researchers of this study found that AAV did integrate into the host genome. This integration can lead to concatemers, inversions, and regions of genomic instability. This has been shown both in mice and non-human primates. The accuracy and read length of HiFi sequencing allows to capture these insertion outcomes in a more comprehensive manner than other technologies would allow you to. We understand that AAV vectors are frequently used to deliver gene editing tools such as CRISPR-Cas9. HiFi sequencing also enables you to more comprehensively validate gene editing techniques. You can use either amplicon-based sequencing or amplification-free approaches on either the Sequel IIe or Revio to characterize CRISPR-Cas9 editing outcomes. This allows you to detect and accurately measure the efficiency of both on and off target effects, conduct insertion site analysis in greater detail, and help assess the potential safety of resulting constructs. Here are some examples of customers using long amplicon sequencing to characterize the potential outcomes of their gene editing approaches. They find that CRISPR-Cas9 editing can lead to unintended effects like large-scale deletions and insertions, structural changes, and complex rearrangements. The read length and accuracy of HiFi sequencing is optimal to enable a more complete assessment of on and off target effects. If you're a researcher in the gene and cell therapy field, you may be aware that next week is the annual meeting of the American Society for Gene and Cell Therapy in Baltimore. We'll be there with a booth and would love to see you there. Stop by booth 728 to meet with us and learn more. Next, I will pass it over to Ervin Garza for his part of the presentation. Thank you very much.
So yeah, I just wanted to say thank you and start out by saying thank you to Nadia and thank you everyone at PacBio for the opportunity to share our work that was published this past summer in Genes. And so my name is Ervin Garza and I am a second year graduate student in Dr. Stephen Gray and Rachel Bailey's labs at UT Southwestern. I am in the Genetics Development and Disease program within the Basic Biomedical Sciences umbrella program. And our work is really going to cover and highlight a lot of the utility that smart sequencing has enabled us to do to engineer AAVs. And so I wanted to start out with just explaining how basic AAV gene therapy works. And in principle, when we know that the disease at hand is caused by a loss of function protein or a gene that's been mutated and renders a non-functional protein, what we can simply do is actually package a functional copy of the gene into adeno-associated virus, or AAV, and deliver it to the patient, where once it's infected in the cells, it actually produces a functional copy of the protein and restoring the normal function of the cell. And in some cases, some diseases actually are caused by a single mutation in a gene, therefore giving them this toxic gain of function disease phenotype. And what we've also been able to do is package artificial microRNAs and take advantage of RNAi technology to suppress or reduce the toxic gain of function protein. And with the advancements in CRISPR-Cas technology, we've also leveraged Cas12 to be able to do this in a much more efficient manner. But what I think is more exciting is actually with Cas9 and prime editing technologies, we're also able to correct the underlying template or the DNA of the disease, potentially fixing the underlying cause. And so AAV was first discovered as what I like to refer to as gene therapy's happy accident, when a couple of researchers were actually making adenovirus, and they found this tiny little contaminant known as AAV within their adenoviral preps. And since then, there's been this explosion of clinical trials where currently now there are hundreds and hundreds of clinical trials utilizing AAV. But what I find also surprising is that only 5 of them are currently approved by the FDA in the US. And so that really renders the question, why is there such little FDA approval, and what's really holding it back? And so some of the concerns and limitations, it's been pretty well established now that with high doses of AAV or undesirable biodistribution of the vector, there is this induced hepatotoxicity or liver toxicity, as well as DRG toxicity when a lot of the AAV gets into the dorsal root ganglion. Additionally, more centered around AAV engineering is actually the species-specific host pathogens interaction, where as groups are engineering adeno-associated virus, we've found that some engineered capsids have specificity to specific species, and therefore rendering them not as translationally useful in larger mammal studies or even humans, such as the case for PHPV. Additionally, even with the current FDA approval and hundreds of clinical trials ongoing, the successful use of AAV is still limited by the amount of cells we're able to target with even the maximal feasible doses with current capsid technology. And so just to outline here some of the FDA approved gene therapies, you can notice right away that we're delivering very, very high titers of the AAV gene therapy. And additionally, this is causing a burden on the manufacturing end, as well as a burden on individuals that need these gene therapies by making them exorbitantly expensive. And so many groups have undertaken this challenge in order to engineer AAV. But before we dive into it, I really just want to take a moment to appreciate all of the clever different ways that people have gone about engineering AAVs, such as peptide insertions that have shown promising outcomes for research, such as PHPV, as I stated earlier. And I know there are some companies that are now starting to see some effective capsids through this methodology. But just to take a brief moment, I want to cover a little bit about AAV genetics and basic AAV biology. And so in simple terms, AAV is actually this single-stranded DNA virus that encodes for 2 major open reading frames known as rep and cap. And so rep is responsible for encoding all of the necessary replication proteins that the virus needs during viral replication. And then cap is the second major open reading frame that encodes for the 3 major viral proteins that make up the composition of the viral capsid. And so these are VP1, VP2, VP3, and they all seem to share this VP common region that makes up about 90% of the virus. And they differ by these small N-terminal extensions as you can see here on the diagram. Additionally, what I find interesting is that AAV also encodes 2 accessory proteins, but these are found in an alternate open reading frame, and these are AAP and MAAP. And just as a reference point, CAP, the gene, actually is about 2.2 kilobases in size, and therefore this is one major hurdle that groups have struggled with just to try and mutate and identify these engineered capsids in a high throughput fashion. And so in the Gray Lab, what we've been able to do is we generate these highly diverse capsid libraries, starting with capsid shuffling, which is what the paper really focuses on. And to give a brief background on what capsid shuffling is, we've essentially taken wild-type AAVs as well as engineered AAV cap genes. We've isolated them and digested them into millions of tiny little fragments. And then with some clever molecular cloning techniques, we've reassembled these capsid genes in these capsid shuffled formats. And we've recloned them back into the AAV backbone, where we then generate our libraries. And so to just establish the paradigm that we in the Gray Lab use to screen a lot of these libraries, starting with the library construction, we start out by actually producing these AAV libraries in a triple transfection fashion, where one plasmid encodes for the ITR AAV library. The second is actually RepCap2, and this is so that we can package all of our AAV library into a common RepCap2 or AAV2 capsid particle. And then the third is the adeno helper that encodes for the proteins that are necessary for AAV assembly and replication. In this first round of purification, we actually harvest what we call our shuttle virus. And so this AAV shuttle virus is then used in a second round of co-infections with adenovirus to produce our final AAV library. And so we're able to titer the shuttle virus at a low MOI so that we can target 1 viral particle per cell. And this is to avoid any sort of mosaicism that could occur if multiple copies of a viral particle infect a single cell. And so once we've taken this final library is what we inject into non-human primates and mice. And then after a few weeks of infection, we actually collect every single tissue from the animal. And depending on the paradigm, our current strategy is actually isolating the nuclei, and we'll go over that later in the talk as to why we focus on doing this. But we essentially PCR amplify the cap gene out of the tissues, and then these cap genes are processed for the smart cell library preparation. And so once we sequence these amplicons from each individual tissue, we can demultiplex them in a high throughput fashion, where we can identify which capsids are enriched in individual tissues. And so just to give a brief introduction or background for smart sequencing, as you probably all well know now, is single molecule real-time sequencing. And so what we've basically done is we take our amplicons of cap genes that are derived from tissues injected with our library. We anneal these smart bell adapters, and then these libraries of our smart cell amplicons, or smart library amplicons, are applied to the smart cell. And what's so great about this smart cell is that it actually has millions of these tiny zero-mode waveguides, where at the bottom is an immobilized polymerase. And this immobilized polymerase is able to capture 1 molecule of DNA, or in our case, it's a single amplicon of a cap gene. And when we introduce this into the sequencing reaction with their phospholink nucleotides, we can actually record in a movie the actual sequencing reaction in real time. And so what this allows is for high sensitivity and actually long read sequencing to occur. And this is what enables us to be able to read the full 2.2 kb cap gene within our studies. And so now I really just want to dive into the paper. where we've essentially started with screening our libraries, as I stated before, into mice and non-human primates. They were injected intrathecal lumbar puncture directly to target the CNS. A couple weeks after injection of the library, we actually collected tissues from the animals and isolated bulk DNA. And so out of this bulk DNA that was extracted from the tissues, we actually amplified the CAP gene via PCR, and then followed the process, as I stated before, with smart sequencing. And then in a second round of injections, we actually made tissue-derived libraries out of the CAP genes that were amplified from the previous rounds of injection. And so this is where we implemented directed evolution to identify capsids that were enriched in the opposing species in the same fashion as we did in the first round. And so, for instance, in this case, where the mouse was injected and we extracted DNA sequences out of the brain and the spinal cord, we actually made this library out of capsid sequences that were derived from the brain and spinal cord, and we injected that brain spinal cord derived library into non-human primates, the opposing species. And we followed that same fashion where we collected the CNS tissues. And in the second round of isolation, what we actually did is we isolated the nuclei from the tissue, and this allowed for increased stringency, and we'll go over that in a couple slides. But it increased the stringency of selection so that we could select for capsids that made it successfully to the nucleus. And once again, we utilized PCR to amplify the CAP gene and smart sequencing to identify which capsids were specifically enriched in those tissues. And then in the second panel of the figure, what we actually have depicted here is the amplicon, or the DNA sequences that we extract out of tissues. Through PCR, we get this amplification of the CAP gene, and then the SmartBell preparation libraries that get put through the sequencer. And this is just a very simplistic manner, or a simplistic diagram to show that with our PacBio data, we just followed this regiment of bioinformatics pipeline, and we're continuing to refine this pipeline to be able to be more efficient and quicker about our analysis. But basically, we take the PacBio data, we do some quality control. We've converted in this paper from subreads to CCS, and then identified each individual capsid sequence's DNA frequency. And then from these frequency, we collapse them into unique proteins. So essentially converting DNA to protein, and then we applied our filter and plotted for all of the figures you see in the future slides. And so this is just an example, a representative example of the starting input library that we injected into the animals, where we first started with analyzing the composition of going in. And so what we've done here is we actually compared to what percentage each individual parent library was similar to the variants in the original input. And so we can start to identify what sequences or what parents seem to be overrepresented going into the animals. And so with the first round of injections, what we're able to really highlight here is how useful smart sequencing is due to the efficiency of analysis. And so in this case, we analyzed the tissues that were derived from the NHP, as you see in the green text, and then the tissues that were, or the capsids that were derived out of the mouse in the orange text. And you can start to see some of the statistics that we get from analyzing the total DNA counts. And then these were converted into unique DNA counts, essentially to filter out any redundant capsid DNA sequences. And then we can start to get a sense of how many capsids we're pulling out of each individual tissue from our input library. And then from this, we convert the DNA counts into unique protein counts to get a sense of how many unique protein capsid variants we're pulling from each individual tissues. And we see quite a broad range of capsids that are pulled out of tissues. And so these were all injected lumbar puncture. So we tend to see the highest amount of capsid variants in the lumbar DRG. But for some reason, this is not the true case with the lumbar spinal cord. And then since the virus essentially has to travel through the CSF and out into the periphery, we actually see the least amount of capsid variants in the deep cortex. So this could just be due to the trafficking of the virus or inability of the virus variants to be able to penetrate through the brain parenchyma. And so what we then were curious about was, do any of the tissue-specific capsid sequences correlate with each other across species or even within species? And so we generated this heat map, essentially looking at the capsids that were pulled out of each individual sample that was run on the smart sequencing side of things. And we found that, for example, there was high correlation with capsid variants that were pulled out of the mouse brain and the mouse spinal cord with those out of NHP CNS, such as the spinal cord of the NHP or even the cervical DRG. And additionally, we can start to see that there are some peripheral organs that seem to have commonalities with the mouse brain and spinal cord. And so in this case, there could be just promiscuous sequences that made it to the spleen, but were also able to penetrate or transduce the mouse brain and spinal cord. And so I think this gives us a lot of information to work with. And so with this study, what we actually started with was just pulling out a couple of capsid variants from each individual tissue, where P1 was highest enriched in the liver, P126 was actually in the mouse spinal cord and the mouse brain. We have P19 that was in the NHP brainstem, P2 in the cervical DRG, and this allows for a much more efficient analysis of the screening process. What I was then curious about was, what out of these sequences that we pulled, what are the commonalities, or what parent sequences do they most similarly resemble? And so in this case, we see that by performing multiple sequence alignments and then looking at a phylogenetic tree or a dendrogram, we can see that P126 has the closest similarity to AAV1 and AAV6, whereas 295 resembles our clone 83 original parent engineered capsid. P72 is actually most similar to AAV8, and 2902 is most similar to AAV2. And then this is where I get really excited as a graduate student in the lab, where I am most curious about the mechanism and really the structural format of these variants. And so by looking at multiple sequence alignments of our hits alongside the parent capsids, we can start to identify in blue the most conserved regions of the capsid, where once we look at it from a biological standpoint, it starts to make a lot of sense, where a lot of the conserved region is, for instance, in this calcium binding domain, or even in the parvo coat, most structural important regions, such as the beta A, beta B. But we see the most variability in the variable regions, which we innately would have expected to begin with. But what we found interesting was, for example, variable region 3 seemed to be highly conserved across all AAV capsids. And so this could give us some indication as to which variable regions may be most interesting moving forward, or even trying to design from a more rational standpoint, but also what regions we should really stay clear from. And so from these lead candidates, we really just wanted to see how well our analysis was performing. And so from the first round of injections, we pulled these 4 capsid variants, P72, P126, P295, and P2902. We actually packaged a GFP-containing genome. So it's the strong, ubiquitous promoter that drives the expression of GFP. And we injected them into mice, intrathecal lumbar puncture, where 3 weeks post-injection, we collected the tissue and actually stained for GFP to look at the distribution of the virus in immunohistochemistry. And so from quantifying the images, what we found is that with one of the lessons we learned is actually that when we're validating some of these capsids, we should probably start with a slightly higher titer. But this allowed for sensitivity at the lower ranges of dynamic range. And so we see that a lot of these tend to perform similar to AAV9, which is the current benchmark for CNS transduction or CNS gene delivery. But we also seem to fall short in some regions, such as this subcortex or in this B region, or even in further more distal regions, such as the cortex or the cerebellum. But there are some interesting findings. For instance, this P2902 seemed to do really well in the cerebellum, but perform poorly in the spinal cord. And so this was more of a proof of concept approach. So we also were interested in looking at peripheral tissues, where the lead candidates were assessed in the peripheral organs. And so we see that there's actually quite a high amount of transduction within the lung, the kidney, the spleen, and the liver. But I think this allowed us to learn a lot of important information. And so I just really want to finish up the paper here with what we learned and what I think is really the most important aspects of this selection, on top of understanding how well smart sequencing enabled us to do these screens. But in the first round of selection, what we found by analyzing bulk DNA is that there was this bias towards AAV1. And these AAV1 sequences tended to perform poorly in the CNS. But when we actually extracted out of nuclei and increased that stringency for capsids that successfully traffic to the nucleus, what we actually found is that we start selecting for capsids that seem to be functionally more relevant, such as capsids that resembled closer to AAV9, or even this clone right here, engineered virus, or clone 83. And this was the information that we gathered from the mice. And the same thing was observed when we analyzed in that same fashion the tissues derived out of the non-human primates. And so in the original library, we see going into the first round, we have this high bias towards AAV1 again. But when we inject the mouse brain-derived library into the second round of screens, we actually see that we start to have more similarities for AAV9 or clone 83, which I think in future studies will render them much more functional. And so just to kind of give a recap, I think with our capsid hunting paradigm that I've outlined here, we can get a sense of, in the manufacturing side of the library construction, a sense of what capsids potentially manufacture better. And so this could allow us to select for capsids that are easier to produce in culture. And then additionally, what we're able to highlight through our screens in parallel within both mice and non-human primates is we're able to analyze for capsids that have higher transduction in the CNS. We could potentially look at capsids that were detargeting the liver. If we adjust the parameters of our screen, we can actually start looking at evasion of pre-existing neutralizing antibodies, or even additionally, the capsids that have enhanced performance for blood-brain barrier penetrance. And then where I'm really excited is to start analyzing in a much more specific manner how we can identify capsids that are more cell type specific or even more tissue specific. And so I'm just going to give a small little teaser here, but this is some work that I'll be presenting at this upcoming ASGCT, where again, just to highlight within our AAV production library paradigm, when we look at the final AAV library, we can see what capsid variants are overrepresented. And so in this case, we can compare them to AAV9. And so potentially this P2 and P51 may have enrichment over AAV9. Additionally, what we've added to the analysis is actually a gene enrichment. So if we focus on the amount of frequency from one library analysis or one sample, we can start to identify enrichment over samples. And so in our shuttle library, we can see what is overrepresented in the plasmid library. But when we use this to create our final library, we can get a sense of what is better at producing. So for example, this P2 and P51 may harbor mutations or sequences that are enhancing for viral replication or viral production. And then additionally, along with some of the work that I've collaborated with some friends in the labs, we've started understanding more from a structural standpoint by reconstructing one of our lead candidates, this P2, And this is just to show that we can validate and find hits that are much more functionally relevant. So in this case, we're showing that this P2 variant that I showed in the previous screen is indeed producing virus better than AAV9. And then specifically in our newer rounds of screens, we were able to identify variants that are performing potentially better than AAV9 with this new analysis paradigm. Likewise, we could do this for the mice, and we can analyze them in multiple administration routes. But likewise, we can start to see that, for example, this P2 variant performs much better than AAV9. But then there, again, coming back to the limitation where some variants seem to perform better in mice than they do in non-human primates, such as this P5, P6, and P7. And then some more exploratory work, but what I'm really excited about is that I've started incorporating and implementing computational analysis into our AAV screening paradigm to really understand mechanistically how these variants are performing better. And so this is just an example of some of the work I did during a rotation in a neighboring lab, looking at intramolecular interactions of the viral protein, as well as intermolecular interactions within the capsid. And then we validate some of these hits using cryo-EM and understanding really from a structural standpoint how we're enhancing the performance of these variants. And so just to summarize the talk here, smart sequencing really has enabled us to screen our capsid libraries in this high throughput fashion, and really emphasizes the importance of whole capsid modifications over small capsid permutations. Additionally, this bioinformatic analysis is critical in efficiently and effectively identifying capsids that are important functionally moving forward. What I really want to stress is from the publication is that we found that isolating nuclei is important for our screening process in order to identify capsids that are functionally relevant. And then the whole context of how you screen your libraries really matters. So for those that are interested in doing a lot of viral or AAV engineering, really putting some thought into how you screen your libraries can really go a long way. And then so this is a little bit more cheesy, but multi-layered problems require multifaceted problem solving. And so this is where I get really excited and happy that I get to work with all my friends, incorporating a lot of different aspects of science together to really solve this complex Rubik's Cube of a virus. And so with that, I just want to acknowledge a lot of important people that have helped me up to this point, specifically my mentors, Stephen Gray and Rachel Bailey, with the tremendous amount of patience and support that they've given me. Members of the Gray Lab that really contributed to all this work, specifically Casey that did a lot of the work on the publication, and then a lot of the current members that are currently helping move this project along with me. Members of the Rachel Bailey's lab that just are there when I'm having my own personal struggles or want to hear me vent, or if I find something exciting, they're also there for the support. Additionally, my thesis committee, Vinny Tagliabracci, Łukasz Jakimiak, Jen Kohler, Kevin Forsberg, and Brett Evers, they've been super supportive. Members of the Viral Vector Core for all the work that they've contributed to producing some of these lead candidates that I'm excited to really share at this upcoming ASGCT, as well as the Bioinformatics Core. They've really had to learn a lot about smart sequencing, especially since this wasn't their original wheelhouse. Our collaborators at Tulane that really made the non-human primate studies feasible and accessible. And specifically at PacBio, you guys have been super helpful technically, and now with the Smart AAV grant, it's enabled me to be able to show a lot of this work at this upcoming meeting. But then additionally, there's a bunch of other people that have contributed to the work that I presented and a lot of the work that I've worked on during my first 2 years as a grad student. And then additionally, my funding sources, I have a fellowship from the Peter O'Donnell Brain Institute, a training grant from NIH, the PacBio Emerging AAV STAR grant, and then most recently, I was awarded the Paul D. Wellstone Muscular Dystrophy Cooperative Research Center grant for discovering myotropic capsids. And so with that, I'd like to open it up to questions and just want to say thank you for giving me your time today.
That was such a fantastic talk. Thank you so much, Irvin. I really love that your research really goes from, screening the sequence all the way to the structure and the function. So really, really love hearing about it. We do have a few questions from the Q&A. So I'll start with the first one from Wesley. The question is for Irvin. Did you perform non-amplification QC sequencing on the various AAVs you generated from shuffling before injection? Wondering what the purity of each shuffled AAV sample would be, or if you see variability within a shuffled capsid type.
Yes, that's a good question. In terms of QC, I'm not sure if you mean what the full sequence, kind of like what was talked about in the introduction, where you can look at the composition of the packaged capsid is, or if you mean individual variants and what their representation within our input libraries are. But just to kind of cover both bases, we did not perform packaged QC in the individual variants of the input libraries, but there was sequencing of the amplicon sequences from our library. So we know like what capsids are present going in. We just aren't aware of, I guess, how complete packaging there was.
Great, thank you. The next question is, did you use any bioinformatics approaches for integrating multi-omics data to elucidate the molecular mechanisms underlying AAV capsid function and host interactions?
Yeah, this is actually a really good question. I'm actually very interested and currently in the process of incorporating some multi-omic approaches. We didn't do so for the purpose of the paper. I think just to kind of give a reference point, a lot of the work that was from the paper was done in the early years of our platform implementation. But now we're starting to look into ways of designing constructs and screening these so that we can do single cell transcriptomics or spatial transcriptomics alongside with our screening in both species. So that's a fantastic question. I think that would be really useful.
And I think Liz had a follow-up question to that.
To the singles, actually, what the single cell one, or what? I actually wrote you my questions. Which one?
How would single cell sequencing help?
Yes.
So what's your plan for that?
And actually spatial, because I didn't know you also mentioned spatial.
Yeah. No, I think those are all really useful platforms, and I think because they're so new and emerging, it's almost like flashy, but people need to understand how to make the best use of it and not just like make this giant heat map and be like, look, we did spatial. I think in terms of single cell transcriptomics, one thing we can get is high content of capsids that could specifically go to tissue types. And since, I mean, I'm very biased towards the brain, but there's this high heterogeneity of different cell types in the brain, and we're continuing to sequence essentially newer types, even subtypes of glia, for example. And so when it comes to really understanding how to develop a newer vehicle to get our therapeutics to the patients that need it most, I think having a hardcore understanding of, okay, this disease is really caused by deficiency in glia or in deficiency of these subtypes of neurons, then we can tailor our capsids to be more favoring towards those specific cell types. So having like spatial and single cell transcriptomics information could help really move the needle forward for specificity, not just tissue type, but cell type as well.
That's amazing. Here's another question for you, Irvin. Chris asks, thank you for the nice presentation. He has two questions. First, how did you do tissue nuclei isolation? And second, does PacBio help build up the procedure or protocol?
So for the nuclei isolation, We have a really handy protocol from a neighboring lab here on campus. Her name is Genevieve Konopka, and she does a lot of single nuclei RNA-seq work. I don't know if we can maybe pin that into the chat, but or you can reach out to my email, and I'm more than happy to share the single nuclei isolation protocol.
And what about the second part? Does PacBio help build up the procedure protocol?
So I'm just going to preface this with you guys do not pay me, but I love you guys so much. PacBio is super helpful in every single question that I've had from like the bioinformatics to the actual library construction or even like how many smart cells do I need to run this many samples? Um, I think depending on your application, there could be some discussions on that. In terms of how we were building our libraries, it was kind of like a trial run method. So we're continuing to kind of improve on that. And I think PacBio does help with the essentially how to plan your experiments.
Great. Thank you. I, I appreciate you speaking so positively without being paid. um, uh, there's another question. So this one is from, I hope I'm going to pronounce this correctly, Xingwei. And the question is, can you leverage more about the bioinformatics pipeline that you use for the PacBio sequencing data from the extracted tissue DNA of your AAV library screen? So I think it's about, telling us a bit more about it. What did you do? What did you use?
Yeah, I'm trying to understand the word leverage as well.
Yeah, I guess maybe in terms, maybe describe a little more in detail what the process is. I think, yeah,
I think so.
that brief diagram really does a really good work of that. But essentially, we're taking um, the raw files that we get from the sequencer and then essentially converting them into FASTA or FASTQ. And then we're using very basic like Python code and bioinformatics tools to extract our like forward primer and reverse primer to constrict only the sequences and sequence length that we want. And then from that, we're essentially just doing programming basics to say, okay, count the table size that we have and give us how many variances in that input, and then convert that into like unique only sequences, and then translate into protein. And then from that, it really just boils down to, I mean, I'm still continuing to improve on how we're analyzing that data now. But it's just making things so that I'm not having to open up this giant Excel table or go line by line, which could take me probably another 8 years of my PhD.
Well, speaking of things that would make your life easier, how AI and machine learning is all the hype right now. So here's a question from Nishat. Thank you for the brilliant research and presentation. Are there computational models or machine learning approaches used to predict AAV capsid properties or behavior based on the sequencing data? Do you know of any? Have you used any?
So, well, this will be another spoiler. I'm actually now working on a machine learning model so that we can implement our data sets that we get from all these PacBio screens. So we are in the process of doing that. I don't have a lot of details yet because we actually have just started like cleaning up the data set, and now we're thinking about what features do we want to feed into these models. Um, but essentially, I mean, I'm not aware of available models that are out there. I think maybe there is one from, like, if I can remember from last year, Ben Deverman's group was doing some small peptide model to predict just these peptide insertions, but I'm not aware of any that are like full whole gene or whole capsid models. And so I think that may also just be a huge challenge or problem to really tackle because it's with a lot of the in silico and computational approaches, we have to find better ways of doing these modeling and simulations. And so I think they do really well with small peptides or small proteins, but it becomes harder as the protein amino acid residue length gets longer. But I think again, the devil's in the detail, and this is where I think I pride myself on that. I like to be very, very particular and nitpicky about details. So I'm hopeful that maybe within the next few years we can have something that is on GitHub or something.
That sounds fantastic. I think people would love to have something like that. And then about being, detail oriented and the devil's in the details. Here's a very detailed question from Ying, who asked, after cap shuffling in viral infection, how do you design the PCR primers for the amplification of the cap DNA to capture all the shuffling population?
Right. Yeah. And so at least within our design, we've cut from start to end. Essentially, we have restriction enzymes that we can cut and isolate these sequences, or even just if they're in plasmids, we can have the plasmid in a PCR reaction. But we maintain the backbone, the ITR backbone, which is essentially like an AAV2, a wild type AAV2 backbone, and we've designed primers that sit just outside of the cap sequence. So if you design the primers just right and you play around with the amplification protocol, you can really optimize how well those amplify out of tissues. Because I guess another problem too is if you're not injecting enough capsids, or if enough viral particles don't get to a specific tissue, it may be hard to detect or even amplify those amplicons, which is where I think, I'm not sure if this could be a useful tool, but I noticed in your introduction, Twist is doing these like amplicon enrichment. So that's something that now I'm kind of curious to look into as well, because that is a challenge.
Yeah, and we'd be happy to work with you on that. A question for Liz came up. What are the criteria? Yeah, what are the criteria to generate CCS from subread? Are there any differences between the different sequencers, so the sequel 2 versus sequel 2E?
I was about to type the answers, but I'll say it is there are no differences. So CCS, and AKA the algorithm that's used to generate the HiFi reads, is what we use and recommend for all our PacBio applications, not just limited to capsid or amplicon AAV sequencing. Now, for those of you who are a little more knowledgeable on how PacBio sequencing works, it's a circular consensus sequencing, right? So each of those passes, people call them subreads. And then the consensus of what the actual molecule is based on those subreads is called circular consensus read, or HiFi reads if they're over QV30. The difference on the sequencer is simply that for sequel 2 versus 2E, the CCS is generated on instrument for the 2E, whereas on the sequel 2, the software, you have to also ask it to run CCS separately after the subreads are first generated. So it's more of a convenience. Sequel 2 is a newer machine, and actually Revio is our latest long-read sequencer, but sequel 2 is the one where we started having on-instrument CCS generation. Irvin, you have a question?
Yeah, so in terms of the HiFi versus CCS, you said the HiFi already has these, essentially if it's Q30 or higher, you can run the HiFi and it'll do that for you. Can you manipulate the Q?
Yeah, okay. So HiFi is a definition, like it's a subset of CCS reads that are over QV30, because not all circular consensus reads will reach QV30 for various reasons. The original read length was too short, or the signals wasn't like as good, right? We have algorithms that determine what's going to be a high quality read. I think your question is like, can I manipulate that? No, by default, the instrument will say these are the QV30 reads. However, if you are an instrument user, you can actually look at the CCS report. It actually will say how many reads are at QV 10, 20, 30, 40, 50, right? So, and then it will say, but I'm only outputting the QV 30 and above reads. There are instances for which under QV 30 reads have been used by customers. I always say, the reason we want people to use QV30 is we know that they're very reliable, easily mappable, but there will be times and certain applications for which the under QV30 reads are desired, and they do get output. It's not like the default, but you can find them.
Great, thank you both. Okay, last question from Christopher. He's asking, you tend to focus on the brain, Irvin. What are your thoughts about the liver?
I hate it. No, I mean, I think if you're someone who doesn't want to get things to the liver, or you're focused on the brain or the muscle even, you'll really learn to love it's a love-hate relationship with the liver. My liver does a great job, but no, I think it's an interesting question because if we know certain properties, like my thought is, if we know certain properties on the capsid surface are getting these capsids to the liver more often, are there these inherent properties that we can't see with our human eye, um, that there's some pattern to it that makes them go better to the liver? And can we abolish those properties so that we can detarget the liver? However, I know there are diseases that are associated with the liver. So actually, if we can get equally better targeting of the liver, there may be uses for that as well, like hemophilia.
Right. Amazing. Thank you. And to close, I would like to thank both Irvin and Liz for their time today and for sharing their expertise and experience. Irvin is a Smart Grant winner for PacBio. We currently have another Smart Grant open. I put the link to it in the chat for you if you're interested in applying this round. And just as a reminder, immediately following this webinar, you'll receive a brief questionnaire. So please do take the time to fill it out. And if you are attending ASGCT in Baltimore next week, please stop by our booth. It's number 728 to say hi, learn more, and win prizes. So thanks again for joining us today. I hope you'll join us again in a future webinar. And with that, take care and have a great day.